Very good evening students, this is your Prasanna Ma'am from NEET World. In this session we are going to deal with plant growth regulators, phytohormones or plant hormones. So this is very important with respect to both NEET and EAP set. Definitely you will get one question every year from this session, or in the successive years definitely one question will be there from plant growth regulators from the chapter plant growth and development. So yes, plant growth regulators are also called as phytohormones. They are also called as phytohormones, or plant growth regulators are also called as plant hormones. They are also called as what ma? Plant hormones. These are of two types. Plant growth regulators or hormones are of two types. Firstly, tell me, what are plant hormones or phytohormones? What are plant growth regulators? They are small simple organic substances. Small simple organic substances or organic molecules, or you can also say chemical substances or chemical substances other than the nutrients of plant body, other than nutrients, and they show drastic or wide amount of the physiological activities. They control all the physiological activities in a plant body. Plant hormones, phytohormones or plant growth regulators are nothing but they are small simple organic molecules or chemical substances other than nutrients in the plant body that provide a wide range of physiological, or they control the wide range of physiological activities in a plant body. As such, plant growth regulators are of two types. One is plant growth promoters, and the second type is plant growth inhibitors. Plant growth promoters and plant growth inhibitors. Plant growth promoters include hormones like auxin, gibberellins and cytokinins. They help in cell division, cell elongation, cell proliferation, differentiation, flowering and fruiting formation is done by plant growth promoters. And second set of hormones include plant growth inhibitors, which include the hormones like abscisic acid, which is also called as stress hormone. They mediate the activities like abscission and dormancy. They mediate the activities like abscission and dormancy. Clear? And there is one more hormone called as ethylene. Ethylene is one more hormone, wherein most of the times it will promote the activity of inhibition and sometimes it will promote the activity of a plant growth promoter. Sometimes it exhibit the activities of inhibition, sometimes it exhibit the activity of promoters. So yes, let us get started with each and every hormone. From each and every hormone, what you have to learn? First is their discovery. You have to know their discovery. And then second is their precursor. And what are the natural and synthetic hormones of that type? And then you have to know what are the uses of that hormone, what are its applications and functions. First hormone if we take, that is auxin. Auxin is discovered firstly Charles Darwin and Francis Darwin. They have applied, they have conducted phototrophic movement. on coleoptiles of seedlings, okay, and they have observed, they have passed on the unilateral light, they have passed the unilateral light on the coleoptiles, they have passed the unilateral light on the coleoptiles, and they have observed that the bending of the coleoptiles towards the sunlight, bending of the coleoptiles towards the sunlight, showing the phototrophic movement. What are the coleoptiles? In a monocot, when we are learning monocot seed, we have learned, right, if you have taken a monocot seed, if you have taken a, what are coleoptiles beta? If you have taken a monocot seed, in monocot seed at the endospermic, at the embryonal region of embryonal axis, you can see the protective covering sheets above the radical, which is called as coleorhiza, above the radical, which is coleorhiza, and above the plumule, it is called as coleoptile. From an oat seedling, when this coleoptile is taken and given a unilateral light, it has shown or bent towards the light, showing the phototrophic movement. This is given by Charles Darwin and Francis Darwin. And next, if you observe, this auxin is first isolated by F.W. Wendt in oat seedlings. F.W. Wendt in what ma? Oat seedlings. What did he do? He has isolated the coleoptiles of oat seedlings, and he has placed this oat seedling, on an agar block, Later, from this decapitated coleoptile, he has placed this agar block, and then this coleoptile has shown this movement again, okay, wherein he said that the active substances from this apical part of the coleoptile are absorbed by this agar block, and when this agar block is placed over the decapitated coleoptile, this has shown the movement towards the agar block. If agar block is placed over this side, this has shown the coleoptile movement is shown towards this side, okay, showing that the active substance, some active substance is present at the tip of the coleoptile, and that active substance is showing the trophic movement or growth of the shoot apex or the coleoptile. This is what is said by F.W. Wendt by conducting an experiment on oat coleoptile, oat seedlings, coleoptile of oat seedlings, okay. And then from human urine, auxin is isolated as IAA, indole acetic acid, okay. This auxin is metabolically inactive in the human cells, that is why this is excreted in the form of urine, and from human urine it is first isolated as IAA, okay. Auxin is isolated from human urine, and it is isolated by F.W. Wendt from oat seedling, where he has conducted an experiment using agar block, clear. And Charles Darwin and Francis Darwin has shown the phototrophic movement of the oat seedlings, clear. This is all about the discovery of the auxin. Coming to the types of the auxin, what are different types of auxin? We can find two types beta. One is natural auxins and the second type is synthetic auxin. Natural auxin and synthetic auxin. Natural auxin, if you see, we have two types of natural auxin. Those are indole acetic acid, indole butyric acid. These are the natural types of auxins. If you see synthetic auxin, naphthalene acetic acid and 2,4-D, 2,4-dichlorophenoxyacetic acid and 2,4,5-trichlorophenoxyacetic acid. These are the synthetic auxins that we use, and the natural auxins are indole acetic acid and indole butyric acid. Where does this auxin occur in a plant body? Auxin occurs in the apical regions. They occur in where beta? Apical regions. Auxin is also called as rooting hormone. Auxin is also called as rooting hormone. It is because it is synthesized in the shoot apical meristem, but most of its activity is shown in the root apical regions. That is why it is called as rooting hormone. Clear? I hope the discovery part, phototrophic movement and isolation, and then types of auxins, presence or the occurrence of auxins in the plant region is clear to you. Now, if we look into the applications and uses of the auxins, what are the uses of the auxins? We will see first. The very first point that you have to remember immediately when you listen the phytohormone auxin is apical dominance. What is this apical dominance? We all know about the apical bud or the terminal bud and the auxiliary buds, right? Apical bud or the terminal bud and auxiliary buds. We have learnt, right? Where does the auxin present? Mostly in the apical regions. So, the presence of the auxin in the apical region is promoting the growth of the apical buds or the terminal buds and inhibiting the growth of the auxiliary buds. Okay, auxin is inhibiting the growth of lateral or auxiliary buds and it is promoting the growth of terminal or apical bud. This is called as apical dominance. Okay, apical dominance is shown by the auxin because of the presence of auxin in the apical buds. This is inhibiting the growth of auxiliary buds. Second is abscission. Okay, wherever auxin is highly present, their abscission will not take place. Abscission will not take place. So, auxin is more present in the young leaves, young fruits, okay, and young buds. So, they will not fall away from the plant body, whereas less amount of auxin is seen in the ripened fruits, ripened vegetables or ripened senescent leaves. Okay, less auxin concentration is seen, in the senescent leaves and ripened fruits So, this less auxin present in this regions or this areas is promoting the abscission. High the auxin concentration in the young leaves, young flowers and young fruits, it is not promoting the abscission. And next is xylem differentiation. Auxin will promote the xylem differentiation, like formation of secondary xylem or secondary xylar elements are formed based on the auxin. Okay, high the auxin concentration, high will be the xylem differentiation, and the primary activity of auxin is cell differentiation. Primary activity is what beta? Cell differentiation. So, immediately you have to remember what are the uses and functions of the auxin ma? Apical dominance, abscission, high amount of auxin will prevent the abscission. Third is xylem differentiation and fourth is cell division. Then coming to the applications, auxin will promote rooting. Auxin will promote rooting in the stem cutting regions. Okay, in the stem cutting regions, auxin will promote rooting. What type of auxin, synthetic auxins that we have to use? NAA and IBA. This auxin will promote naphthalene acetic acid and indole butyric acid. This type of auxins will promote rooting in the stem cutting regions. Second is flowering. Auxin promotes flowering. What type of auxins will initiate flowering? NAA and 2,4-D. Third is parthenocarpy, that is formation of seedless fruits. Formation of what ma? Seedless fruits. The seedless fruits are formed by IAA and IBA. When we apply IAA or IBA to the plant bodies, seedless fruits are formed. So, auxin is inducing parthenocarpy. Last is weedicides, okay. Auxins like 2,4-D, 2,4-dichlorophenoxyacetic acid, they act as best weedicides and they promote weed free lawns. They promote weed free lawns. They will kill all the weedicides, like mainly dicotyledonous weeds are killed by 2,4-D. Okay. These are the applications of auxins and these are the uses or the functions of auxins. So, what you have to remember with respect to auxin beta, very important, both the applications and uses. What you have to remember, apical dominance, abscission, okay, less the auxin, more the abscission, xylem differentiation, cell division, these are the functions, and most importantly the applications are parthenocarpy, weedicides promoting flowering and rooting in the stem cutting regions. Okay. This is all about auxins. Coming to cytokinin, it is the scientist Skoog, okay, who is the scientist Skoog and his co-workers have isolated the cytokinin, okay, natural form of cytokinin like coconut milk, and Miller et al. Is the scientist who has isolated them from the kernels and from coconut milk and kernels of maize. The natural form of cytokinin obtained is called as zeatin. Do remember beta, very important. What is the natural form of cytokinin? Zeatin. And it is Miller et al. Who has autoclaved herring sperm DNA, herring sperm, that is a fish, okay, herring sperm DNA, and he has obtained the synthetic form of auxin, that is kinetin. Synthetic form of auxin is what ma? Kinetin, okay. And what is the precursor of cytokinin? Precursor of cytokinin is purine or adenine derivative, we will call it as. What is that ma? Adenine derivatives. We will call this adenine derivatives as N6-furfuryl aminopurine. N6-furfuryl aminopurine. Clear? So, these are the precursors of cytokinin. And what is the natural form of cytokinin? Zeatin, which is obtained from kernels of maize, and the natural form of cytokinin is also obtained from coconut milk. It is also obtained from yeast extract. Okay. And Miller et al. He has autoclaved the herring sperm DNA, and what is the synthetic form of auxin? That is kinetin. Synthetic form of auxin is kinetin, and the precursor of auxin are adenine derivatives like N6-furfuryl aminopurine. And the occurrence of auxins mainly include apical regions and mostly apical regions and buds, apical region and shoot buds, which include auxiliary buds, young flowers and fruits. These are the regions in the plant body where cytokinins occur. From here, what you have to remember, what are the natural forms of cytokinin? Coconut milk, yeast extract, kernels of maize, that is zeatin. Miller et al. What is the artificial form or synthetic form of cytokinin? Kinetin is the synthetic form, and the derivatives of cytokinin are adenine, which is N6-furfuryl aminopurine, and the occurrence of cytokinin includes the apical regions like shoot bud, axillary buds, young fruits and flowers. Okay. When it comes to the functions of cytokinin, cytokinin functions mainly include overcoming the apical dominance. Okay. Overcoming the apical dominance. We have observed, right, the auxin which is present at the apical region, it is inhibiting the growth of axillary buds. It is inhibiting the growth of axillary buds. But cytokinins presence in the axillary buds, cytokinins present in the axillary buds, it will counteract the effect of auxin. Cytokinin which is present in the axillary bud, it will counteract the effect of auxin and it will overcome the apical dominance. Thereby, it will promote lateral growth of axillary buds. Thereby high amount of lateral branches are formed. This is the best practice that is used either by cutting off the apical buds or by giving the cytokinins. Mostly it is applied for tea plantations and hedge making. Tea plantations and hedge making. Just a second. Yeah. Tea plantations. This, it is better if we write in the auxin itself. Yeah. Here. Yeah. Tea plantations and hedge making. Best application of auxin. Okay. This is the best application of auxin, that is tea plantations and hedge making. Okay. By decapitating or removing the apical buds region of a plant, we can promote the growth of lateral buds, or we can also give cytokinins to increase the lateral growth of the branches. Second is cytokinins main activity is to delay senescence, where we will call this as Richmond-Lang effect. Richmond-Lang effect. Okay, this is Richmond-Lang effect, where this will delay the senescence. And third is cell division. Okay, cell division is the common function of auxin and cytokinin. Third is growth of leaves and shoots. These are the functions of the auxin. Okay, these are the functions of auxin. When it comes to the applications, by applying cytokinins in the growing regions like growing fruits, flowers and shoot buds, this will overcome the process of senescence, giving you the complete growth. Okay. And in tissue culture, how cytokinins are applied? Both auxin and cytokinin play crucial role in tissue culture. If auxin and cytokinin are applied in the equal ratio, for example 1:1, equal ratio if they are applied in tissue culture, it leads to the formation of callus. Okay. If more amount of auxin and less amount of cytokinin is applied, this leads to root formation or rooting in the plant tissue. Okay. And if there is less auxin and more cytokinin, this will initiate shooting. In the plant tissue culture. Clear? So, you have to remember, if auxin and cytokinin are in the equal ratio, this leads to the formation of callus. If auxin concentration is more and cytokinin concentration is less, this leads to rooting. If auxin is less and cytokinin is more, this leads to shooting. These are the applications of cytokinin and these are the functions. The primary thing you have to remember is both auxin and cytokinin are synergistic. Synergistic means similar in their function at cell division. Okay. Both of them will promote cell division, and Okay, synergistic function of auxin and cytokinin is cell division. Okay, antagonistic function of auxin and cytokinin, both of them are function wise they are opposite to each other, that is apical dominance. Okay, with respect to apical dominance they are antagonistic to each other, wherein auxin is promoting the apical dominance, cytokinin is inhibiting the apical dominance. Clear? So yes, this is all about cytokinin. When it comes to gibberellin, okay, gibberellin, it is discovered by the scientist Kurosawa. Okay, Kurosawa, when the usual size of rice seedling is around like 2 meters, but then it has because of a fungal disease the usual size of a rice seedling is like around 2 meters, but the rice seedling has increased their size up to 20 meters height. Okay, up to 20 meters. Okay, like this. Okay, this increase in the length is because of a fungal infection. This is because of what, ma? Fungal infection. Fungal infection, and the fungus name is Gibberella fujicuroi. Gibberella fujicuroi. This fungus has released some active substance. That active substance is named as gibberellins. The active substance is named as gibberellins. Okay, this gibberellins active substance, this leads to the elongation of the stem axis in the rice seedlings. Okay, this is what is discovered by the scientist Kurosawa. Okay, and the precursor for the hormone gibberellins is acetyl the precursors for gibberellins, just a second. Yes, acetyl CoA and mevalonic acid. Yeah, precursors include acetyl CoA and mevalonic acid. Okay, it includes acetyl CoA and mevalonic acid. Clear? These are the precursors of gibberellins, and the first discovered gibberellins are gibberellic acid 3. This is the first discovered and widely used gibberellin. Okay, gibberellic acid is acidic in nature. Okay, this is acidic in nature. Coming to the applications and uses. Or functions of gibberellins. Gibberellins will promote seed germination. Gibberellins will promote seed germination by promoting or releasing the hormones like amylases, which will degrade the starch which is present, which will cleave the starch which is present in the seed, and it will give the nutrients for the seed to germinate. Okay, gibberellic acid or gibberellins will release the hydrolytic enzymes like amylase that will cleave the starch and carbohydrates which is present in the seeds, and it will give those simpler sugars for the seed germination. This is how gibberellic acid is promoting the seed germination. Second is bolting. Bolting is seen in the rosette plants. Bolting in the rosette plants like cabbage and beetroot. Okay, in these plants like cauliflower, cabbage, beetroot, what happens is prior to the flowering, gibberellic acids or gibberellins will increase the internodal elongation. This will promote what, ma? internodal elongation. This will promote internodal elongation. By promoting the internodal elongation, the length or the stem size increases, and post flowering this will give rise to a very big size of vegetables like cauliflower, cabbage and beetroot. Okay, gibberellins, promotion of gibberellins or gibberellic acid prior to the flowering will increase the internodal elongation and increases the size of the rosette plants post fertilization. For example, cabbage, cauliflower and beetroot, and this will increase the stem elongation, stem size or stem elongation is increased. Best example is sugarcane. Okay. Here are the applications of gibberellins. First is fruit growth. For example, apple. Okay. Increase normal size of apple. If this is this much, this is the normal size of apple. If we apply gibberellins, this will give a proper shape of apple with increased size, Same way, the stalk length of the grape is increased. Based on that, many grapes are formed to that stalk and increase in the fruit size is seen. That is because of the application of gibberellins. And gibberellins will delay ripening. Delaying of ripening is shown by gibberellins. And in brewing industries, to increase the malt yield, gibberellins are added. Okay, gibberellins are used to increase the malt yield in the brewing industries. And next is enhances the sugarcane yield by promoting the stem elongation in the sugarcanes. Then quick maturity is seen in the conifers. Conifers will quickly attain their reproductive phase when we spray gibberellins. Okay, they will quickly attain the reproductive phase. Okay, so this is all about gibberellins, both the applications, functions. Then comes our abscisic acid. Abscisic acid is also known as stress hormone or anti-transparent hormone. Okay. So, yes, abscisic acid is also called as what ma? Stress hormone or anti-transparent hormone. Abscisic acid, okay, abscisic acid is acidic in nature. Abscisic acid is acidic in nature. In the year 1960s, 3 scientists have discovered abscisic acid in 3 different ways, like Dorman, abscissin, just a second. Dorman beta, abscissin 2, and then Dorman beta, abscissin 2, and one more scientist has discovered this as inhibitor beta, and Dorman and abscissin 2. These are 3 different ways in 1960s where abscisic acid is discovered. Later scientists have noticed that all these 3 substances are doing only one function, that is inhibiting the plant growth and stomatal closure, okay, during the stress. The main precursor for abscisic acid is violoxanthin. This is the precursor of abscisic acid, okay, and this is also called as stress hormone or anti-transparent hormone. This is because the abscisic acid is activated or released during the stress conditions of a plant. Whenever plant is in stress condition, abscisic acid is released, and at that time when abscisic acid is released, the very first activity is closure of stomata. The very first activity is stomatal closure. Stomatal closure is seen. And second is seed dormancy. Whenever there are unfavorable conditions outside, seed remains dormant without seed germination. So, seed dormancy is promoted by abscisic acid, and abscission of the older leaves or senescent leaves, older fruits is promoted by abscisic acid, and they will inhibit the metabolism. Cytokinins will promote the protein synthesis, it will promote the metabolic activities, but abscisic acid will inhibit the metabolism. With respect to seed dormancy, abscisic acid is antagonistic to gibberellic acid. Okay, abscisic acid is antagonistic to gibberellic acid with respect to seed dormancy, wherein ABA is promoting seed dormancy, GA is promoting seed germination. Clear? And how abscisic acid is used? Okay, applications include anti-transparent. Applications include what, ma? Anti-transparent. Okay, this will inhibit the process of transpiration, mainly in the desert plants. Applying ABA will promote the plant to overcome the stress conditions. External stress might be, internal stress might be. Abscisic acid will promote the plant's growth or the plant life in the stressful condition. And second, by promoting the dormancy, especially seed dormancy. These are the activities of abscisic acid. Next comes the ethylene. Okay, ethylene. It is the scientist Auxins. He has observed that there is some gaseous release from the ripened oranges. Some gaseous release from ripened oranges. This gas is promoting the unripened bananas to ripen bananas. Okay, unripened bananas to ripen bananas. So, the gaseous release or the volatile gas, we will call it as volatile gas, which is released from ripened oranges, is promoting or ripening the bananas. This is what observed by Auxins. Okay, later that gas is said to be ethylene. Ethylene is present in the occurrence of ethylene is ripened fruits. Okay, ripened fruits. In ripened fruits, ethylene is present. Okay, and the synthetic form of ethylene is ethaphone. Ethaphone is the synthetic form of ethylene, and the precursor of ethylene is, just a second, precursor of ethylene is methionine. Methionine is the precursor of ethylene. These are the basic points that you have to remember. When it comes to the functions and applications of ethylene, first we will look into the functions. Especially in the rice seedlings, this shows triple response. Triple response is the horizontal growth. First is what, ma? Horizontal growth, and swollen axis. Second is swollen or thickened axis, and third is formation of apical hook. Third is what, ma? Formation of apical hook. These are the three activities, that is triple response, which is shown by ethylene mainly in the rice seedlings. And second most important function of ethylene is abscission, that is fall off the ripened fruits. Okay, ripened fruits are fall off from the plant by ethylene, and it will promote the senescence. Third point is it will promote the flowering and female flower formation, especially in cucumber. Okay, in cucumbers they will promote the femaleness of the flowers. Only carpels are developed, not androecium, that is called as female flowers. Okay, it is seen in cucumber, that is promoted by ethylene. Next, stem and petiole elongation in the aquatic plants, especially in some submerged plants and those plants which grow in the wet areas, like rice seedlings, right? Rice seedlings, the petiole elongation and stem axis elongation is seen, that is because of ethylene. And ethylene will promote adventitious root formation in the stem cuttings. Okay, ethylene will promote adventitious root formation in the stem cuttings. Okay, it will promote adventitious root formation in the stem cuttings. Adventitious root formations in the stem cuttings. When it comes to the applications, we spray the synthetic form of ethylene, that is ethafone, for fruit ripening, and ethylene is used to promote the apposition of the older fruits, and then it will increase the fruits and fruitlets. Okay, by promoting the femaleness of the flowers, it will increase the fruits and fruit set. Okay, and sprouting of storage organs like rhizomes and tubers. Okay, ethylene will promote the sprouting of storage organs like rhizomes and tubers. Okay, this is all about ethylene. The main activities you have to remember is triple response, apposition of the older or senescent fruits, and it promotes the femaleness of the flowers, that it promotes the growth of female flowers, for example cucumber. Okay, and it will promote the stem and petiole elongation in the aquatic plants and adventitious root formation over the stems. Okay, this is all the important points with respect to ethylene. This is the final chart. This is the final chart where you can compare all the hormones with their precursors and their chemical composition. Auxin, for example, IA, it is composed of indole compounds, for example indole 3 acetic acid. Precursor is tryptophan. Second, cytokinin, which is the natural form is zeatin, synthetic form is kinetin. These are adenine derivatives like N6-furfuryl aminopurine, and the precursor is adenine. Third, gibberellins. Gibberellins, these are composed of terpenes, and precursor is acetyl-CoA or mevalonic acid. Abscisic acid, the chemical nature is sesquiterpenoid or terpenoids, and the precursor is violoxanthin. Ethylene, the precursor is methionine, and it is a gaseous hormone. Okay, this is all about the phytohormones. In next sessions, we are going to do all the previous year questions from 2019 paper to 2024. So stay tuned. All the best for your preparation.